Okay, so experiment 33 is bacteriophage titrations. And so bacteriophage background. So viruses, as you know, are obligate intracellular parasites that must infect the host cell to replicate. Got that straight out of my manual. And so their composition, every virus has some genetic material, whether it be RNA or DNA, a capsid, and an envelope. And the envelope surrounds the capsid. So it has a very wide range that can infect animals, plants, bacteria, but each specific virus typically can only infect a very specific host. That like it can like only infect like one animal or like maybe two animals or stuff like that. That they're very specific to that organism. And so their life cycle is pretty much as follows. So they attach to the host cell. So whether it be a bacteria, a eukaryotic cell, or whatever it may be, they will then release their nucleic acids into the host cell, and then they will incorporate that nucleic acid into the viral genome, and so then you kind of have two kind of cycles that you can go through. You can go through the lytic cycle, that after you incorporate it, you then start just making your viral components. So you start making your capsid and your envelope, and you replicate your DNA. You make all those things, any proteins that you need. You replicate them, and you put them into like a virus, and then you release those virions. A virion is another word for a virus. But then you release those virions into the environment to go infect more cells. Or you can go through the lysogenic cycle. Lysogeny is where the bacteria just remains in the like nucleus of a cell, or if it's a bacteria cell, it just remains in that chromosome. And then under certain conditions, like if it's exposed to something that might kill it or something like that, it will then enter the lytic cycle. And then in the lytic cycle, it will then go through all this of assembling everything and then releasing into the environment. So those are kind of two. So everything that goes under the lysogenic cycle goes through the lytic cycle to replicate more. It's just some microbes go through that lysogeny before they lyse the cell. So what a bacteriophage is the virus that infects bacteria. And so most of the bacteriophages go through the lytic cycle and without going through lysogeny. Lysogeny is a lot of times just for like eukaryotic cells, whether it be plants or animals. But what we're going to be doing is we're going to be doing a plaque assay. So it's a way to determine how many viruses are in a specific sample, not a cell. It's supposed to say sample here. And we're going to be studying specifically bacteriophage lambda. So bacteriophage lambda infects E. coli cells. So what we did is we had a tube of bacteriophage lambda, all right, and we did a serial dilution, and I have another video on YouTube about serial dilutions with regard to like lytic bacteria, and like this whole bacteriophage titration stuff. But we did a serial dilution on that, and then we mixed E. coli in with soft agar, all right. And then what we did was we took the soft agar, the E. coli, and the dilution that we made, and we mixed it all together, and then we poured it over our solid agar. So you're going to be giving plates that have solid agar already poured, but doesn't have this top one. So you're going to have to be making this that has the soft agar, the lambda, and the E. coli in it. And what that allows is we're going to spread that over the solid agar, and then that allows it to grow. So the solid agar allows the E. coli to grow, and then the phage will then infect the E. coli, and it will then lyse the cell. And when it lyses, it makes what's called a plaque. And it's just the lysis of certain cells. So if you look at our plate right here, each one of these little dots here is a plaque. All right. So and all of the white you see, those are all E. coli growths, but there is no bacteriophage lambda in those cells, so there's no like plaques here. All right, so this is just like E. coli that's just grown and nothing else. So the white is E. coli, the holes are the plaques. All right, and so what you can do is you can count all these, right? And then you do a serial dilution because you want your number to be between 30 and 300. But you're going to count all these, and once you count them all, then you can calculate back to find the concentration of that bacteriophage lambda. And it's a way of like getting concentrations of certain solutions and stuff like that. But it's kind of a complicated experiment with regard to you have to do the titrations, the serial dilutions, and then you have to find. So it's a kind of very interesting experiment, but you can get the plaque forming units per milliliter that way. So the takeaway from this experiment is you need to know the life cycles of a virus with regard to and the lytic cycles and the lysogenic cycles. What makes up a virus? Most of the time it has enzymes in it, but you don't need it. But you need nucleic acid of some sort, whether it be RNA or DNA. You need the capsule and you need the envelope, and then you kind of need to know the experimental setup of the plaques and all that kind of stuff, and then you need to know how to do dilutions, which is in that other video. So watch that other video if you need a little bit of help on there. It's a relatively good video, I think. I made it though, so go watch that video if you don't get intro titrations, and then if that doesn't work, you can go ask your TA or something.